let's get started again. So we're talking about protocols and what they look like. So, this, if you're doing just data analysis, it's fairly straightforward, like I showed you. This is just one example. There are many good protocols out there. By the way, in addition to researchers, or just into the resource I showed you, if you want to learn how to do these things, I learned to write basically by reading papers in JAMA. Not like you put them beside your paper. They're all well written, they're edited, and try to do it like that, right? So anyway, if you want to find good examples, or ask someone for a good example, and just copy them. Or don't. We're talking about plagiarism outside. Don't copy the actual study and the data. Copy their format. Anyway, but this is giving you an idea. We published. This is a trial we're doing. It's about self-management, which is developing skills to be a more effective person, managing your own disease, basically, or working with your healthcare providers to do it. And you can publish these. Many journals will publish protocols now. And just to give you an example, it's a full, it's like a full paper. You have your abstract. You'll see that compared to results publications, there's a lot more here. It's a fairly long, rationale introduction. And there's a lot of detail in the methods, right? How we got our participants. There's a flow chart of how things will get into. And I know you can't digest all this now, but just showing there's a chart for timing when you're collecting each point of data. And you'll see even things like how we're evaluating whether it's feasible, and it's described very carefully. What our interventions are in the comparators, et cetera. You'll even see even how we're going to randomize the outcomes. But you'll get the point. How we're going to randomize has about a third of a page, right? Somewhere. But it really is a recipe at this point. And you'll see that one thing that we have, in a protocol like this too, we put basically it really is a recipe. Anything you would need to do to do our study. So we'll attach. The nice thing about publishing now is there's no limit to what you can put in a supplemental material. Like we in some of our papers, especially like our big meta-analyses on the individual participant data, we have our supplements are like 150 pages sometimes, which is not that you have to do that. And again, if you're smaller projects as a student, you may have a page or two, but we put in our consent forms, we put our data collection forms. Like if you're using established measures, it's very easy. You just describe the measure. No one needs to see it there, right? And you might not be able to put it there depending on the copyright. But if we have any kind of questions that ask, how we ascertain people's whether they had ulcers or not, or anything. If it's not publicly available, we put it in our appendix. We put our intervention material in our manuals. And again, coming back to the transparency and completeness, my salary all comes from your taxes. So I don't believe I have any right to not show you anything we do, right? So you can actually, we have links. You can put links too. We don't actually put a 150-page patient manual. We have a link to the manual, a link to worksheets we do with patients, whatever we have. Our fidelity is how did you deliver the intervention the way you said you were going to deliver it? And we have a protocol for assessing that, and that protocol is back there. The statistical analysis plan, that's a little different than the statistical analyses. And again, this is when you're getting into bigger research, right? But you have a half a page or a page maybe in your manuscript. This is a recipe that another biostatistician could follow somewhere. So, and again, I'm happy to share these with you. I won't show them to you now just for time, but it'll be like 8 or 10 pages sometimes of exactly how variables are coded, what the code is, what the models are. And more actually, once the papers are done, people are putting their code available for people, so someone can actually, run your analysis. We want things to be as transparent and clear and easily checked and verified as possible. And you'll see, there's some other stuff. I don't think we include our standard operating procedures in the protocol, but we actually have these things just to show you. I will show you this one. And again, this is a trial that has, these are more complex things, because this trial has people from scleroderma who are from 6 or 7 countries who are coming in through our cohort at different times. So there's some complexity to it, right? And it's trying to enroll 400 or 500 people. So we're not doing as well as we like. So if you happen to know anyone with scleroderma who wants to be in a trial, no, I'm just kidding. You'll see even, yes? Well, you can use it as a template, yeah, they can see what you've done. But also, a lot of times in our field, like how well someone was blinded, right? An intervention. So it's really hard to blind in non-pharmacological issues anyway. So sometimes it's just, you can't, a lot of our trials aren't blinded. You either got it or you're on a wait list, you know you didn't get the goods, it's not blinded, there's nothing we can do about it. But sometimes it's not as clear. And it really depends what you tell people and exactly how you tell them, right? And consent. That's a lot of trials. The basic key issue is like, do people know they got the goods or not? And that depends on how they're consented. So I think for a lot of our reviews, we often code things as unclear. We don't know if it was blinded properly or not, because we don't really know what they were told in the consent process. And so it plays that role also. But you have it, right? It's easy. So it takes, I don't know, someone 10 minutes to form. Well, it's already formatted. It's a consent form, so you just have to attach it. But this is more of like, this is an SOP or a standard operating procedure. It's just, again, and it's not going to be as complex, but even if you're doing a simple study and collecting your own data for the first time, you might be a page or two, but you want to have an operating procedure. And you'll just see this just tells everyone, what they do. And this is a complex trial, so there's about 6 websites that have logins and, how we recruit the participants and where people put what data in the Excel spreadsheet and how they log in. Because, you have different research assistants and different people doing it. And if something can go wrong at will, I don't lay awake every night thinking about this. But imagine if you go halfway down the trial and your team's doing something in a way you didn't envision, and they don't, people have different backgrounds, so no matter how smart they are, they're not necessarily going to be on the same page about exactly everything and what the implications might be, and they may do something that is going to impact the study. So it's really important to document that stuff as well as possible in your protocols. This is a little bit different. That was a protocol for collecting primary data, right? We're actually going out and collecting data from people. The other area that we work in, I know a lot of people at the University of Montreal work in, is evidence synthesis, right? Systematic reviews, scoping reviews, meta-analyses. It's also really important to have the same kind of protocol, the same thing here. What you include is a little bit different. But again, the supplemental materials are different. And we work with a really, really great information scientist, or librarian, they used to be called, I guess, but who's really advanced. But now, you don't have to just describe your search. This is our search document. And you'll see, you have to develop these things anyway, right? Oh, I'm freezing. Yeah, so you have to develop your search terms anyway. So put it there. No one, sometimes people say, oh, here's my PubMed search, and put all your searches. Someone can use exactly what you have. Why not, right? Someone should be able to find the same. People should be able to disagree with how you defined your research question and how you included, but they shouldn't have troubles with finding what you said you found, right? So give them all the information they need. We include, we do all of our screening for systematic reviews in an online program, but each of them has basically, the title abstract. Exclude it if it's not human data, and here's the instructions. Exclude if it doesn't. Every single, all our manuals that people use to actually select studies for our review, again, they're in the back of the supplements. Our data extraction forms, right? When you're doing a review, you extract data from each study. So how you need a form for what you got. So just put it there. Let people see exactly how you got it. Because sometimes when people only provide a couple paragraphs, we extracted these variables. It's not quite clear. And depending who's going to use your study, you never know what information they might need. And this way, everyone has anything they could need. All right, so what do you do with your protocol, right? So you're developing a protocol. There's a number of ways to do it. So I think at that point, it should be publicly available. So, and again, there's enough shenanigans that go on in research. As a methods reviewer, I review all the time for this particular journal. I want to see those protocol. And and oftentimes they can't come up with a protocol that I can verify was pre-trial. And people change enough stuff along the way, right? You don't want people to develop their methods based on the results, and that happens so much in our field. If you collected 10 outcomes, but only 2 were significant, now we say in our methods we only collected 2, and this happens, I'll show you in a minute, happens how often it happens. You're developing your methods based on what you already found, and by definition, this is not helping our community anyway. In fact, it's misleading, right? So you want to know that there's a protocol a priori. Sometimes, certain, like JAMA review, they ask you, they have to submit their ethics protocol. So those are at least often date stamps. So you can say, okay, it probably is a priori before the study. You can probably use this. But oftentimes it's not so clear. So it's good practice to put something out in the public domain, right? That people can verify. And you can say when you put your study out there, I had a publicly available protocol a priori. Here's where you can access it and you can check what I've done. So, okay. So, but then your 2 options are doing that are basically publishing it, which is becoming what it means to publish versus posting somewhere, which is graying in, as we go forward what that means, the difference is. But so you can, the protocol I showed you was published in a journal, right? And that's one way to do it. You can also just post it on a server somewhere and do that. So what's the difference? I think if it, again, depending on if our stakeholders, who are, it's a big area or waiting to see what you're doing, you probably want to publish your protocol. It's going to be more visible. It's going to get into PubMed. It's going to be found more easily. People know what's out there, right? Do you have money for this? It's not cheap. Where can you publish a protocol in a journal? The one I have grayed out here is a preprint server. Are people familiar with preprint servers? They're basically, you can submit your papers there before they go undergo peer review. And there's advantages and disadvantages, right? In COVID, they got some bad press because stuff would get out, and then it was found to have some real problems. It would get out in the press, right? And once it gets out in the press, it becomes real. But the advantages are that you can actually get evidence out there more quickly, and people can see what's been done. So I didn't realize, but actually this is a nice preprint server where you can put your papers, right? And almost all journals now will let you put it on a preprint server like this one. It's not the only one. And then also submit it for peer review, and they won't say you've published it somewhere else. It's more and more accepted. But they actually now accept protocols, which I didn't realize until I was preparing for this talk. They didn't used to. But you could put your protocol there, and it gets indexed on PubMed. So that actually is free. It's free, and it serves the purpose of getting it out that people can find your protocol, right? It's searchable now. The only thing here, the only thing here as I go down below is that you don't get any review of it, right? You're putting whatever you have. So no one's going to look at it if you want peer review. Other journals that do this, like Trials, the journal Trials does a nice job with protocols where you publish, but they're not cheap, right? It's like something like $2,000 or $3,000, I think it's like $3,000 US. So you've got to really have a budget there. And it may not honestly be justifiable. If you can put a preprint on medRxiv for free, you got to think twice, right? About if you spend your again, it's just like healthcare, right? If you're spending it here, you're not spending it somewhere else. You're not sending students to a congress. You're not doing something else. So Contemporary Clinical Trials is actually a trials journal that doesn't charge. And I had students, I teach a trials class at McGill, and a couple of students last year from the School of Education published their protocol then in this journal because they didn't have funds for it. They got it in. And sometimes some subspecialty journals, like I work in rheumatology. A big rheumatology journal typically won't publish protocols, but a subspecialty that focuses on scleroderma, they've published protocols. They're free. They get peer reviewed. So that's another option. How to decide is, again, do you want peer review? And again, that depends. So like some journals like Trials, if you've actually already been peer reviewed by, like, say, CHR, and that's the one in Canada they'll take, usually a national level body, they won't actually duplicate the peer review, which makes sense, right? At that point, they're considering it wasteful to duplicate it. The preprint servers, of course, won't peer review it. And some journals have better peer review processes than others. I mean, there's a whole other story of what's happening to peer review right now, but people are so overworked and so overburdened that they can't find reviewers. Oftentimes the people reviewing the papers don't have the wherewithal to do it. So sometimes it's better not to get peer reviewed. But that said, some journals do it better. I just know from experience, like Trials does a good job with this, right? You pay for it. They're going to get it reasonably done. Contemporary Clinical Trials, probably less so. But it is what it doesn't charge either and so forth, right? And then, do you have time? It depends on what the time factors are, and you have deadlines and people's moving forward and funding. Peer review can take a long time. Sometimes people publish, one thing I see people do a lot is they actually publish their protocols and they come out a month or two before the final paper comes out. And honestly, other than generating publications, they're generally not very useful, because if they publish the final paper, they could stick the whole protocol in an appendix online. And if we don't get to see it until a month or two ahead of time, they've already seen most of their data or all their data anyway. So we have no way to vet that it actually occurred ahead of time. So I don't think it's that probably doesn't serve any purpose other than getting another publication for somebody, right? But so think about it. The main point is, though, you want it out there where you can come back and say it was out there, publicly available before I started this. Yeah, question in the back? Sorry, my hearing's bad anyway, and there's a fan, and I know I keep coming back, and I keep feeling more and more like whatever. Okay, so, all right. Another good place to put these, I didn't realize that medRxiv had free preprint protocols that were on PubMed, so I think we'll start going there more now, but the Open Science Framework is actually a great place too. And what's really nice about this is, one, you can put your protocol there, but two, you can put all sorts of other documents. So you can put data files there. You can put, for some of our scleroderma interventions, we put patient manuals, instructor manuals, anything anyone would want to use. You can deposit as much information as you want. And so it's a nice, you could also keep them on your own servers and say, but there's no guarantee we're around, and this is a central place that's publicly known, that people can go and get all sorts of files. That's one nice aspect of the Open Science Framework. So we typically put our protocols here, the ones that we don't publish in a journal, like the student protocols, but we'll probably, like I said, I didn't realize that the medRxiv was free until recent, just a few days ago. But we'll probably go there now, and I think, but you could probably put in both places. You publish it there so it gets on PubMed, and if you could also put it here if you think you might have other documents to upload with it. So what do you do when you publish then? How does this all come into your publication? And these are just a couple passages out of our papers, but our first paragraph of our methods always says, we published a protocol, and here's where you can find it. And by the way, the nice thing about any of these things, so if you go on if you publish in a journal, it's going to have a submission date. So people can verify that you submitted this thing before you started. If you publish on medRxiv, the preprint server, same thing. It's dated, right? And if you go on the Open Science Framework, it's also dated. And it also tracks on that site when you've removed any files. So you could put a protocol there in that one and remove it, which you can't in the journal or the preprint. But it'll tell people at least that you've removed a protocol. Right? So if you put up one protocol, it's dated, you're good, and it's there. So that gives you more credibility, right? So if you have people who care about these things reviewing or looking at this, they'll say, okay, I can check what they've done ahead of time. They didn't change their methods to fit the results. Or if you did change your method to fit the results, they'll catch you, but that's probably not what you want, right? And then we almost always have a section of amendments. And this one has more amendments than we might otherwise. But we had a trial in the mental health one in COVID. We literally went in 3 weeks from, we have a problem here, we met with our patients, we got ethics, we got money, we got a protocol in 3 weeks, and we were enrolling people. It was never to be done again. I'm too old for that. But in any case, we had a number of amendments, right? So what were amendments? We put a protocol up online, and then we only had one outcome time point because that's all the money we got. We ended up getting more money. We put another follow-up outcome date. So that goes here. That's an amendment to our protocol, right? We tell people why we change things. You might change things because, the data came out in a way that you didn't expect, and it wasn't appropriate to analyze it as you'd planned. So you change your analysis plan and tell people. So there's no shame in changing it. There's only shame if you change it and don't tell anybody why, because then people are going to think you changed it because you didn't get the result you wanted, right? Because that happens so often. So tell people you have one, and tell people any amendments. Okay? Yeah. Yeah, anything. Like the one I showed you first, the student protocol, I said was data analysis only, not collection. So our graduate students, especially the master's students, only use existing data because they don't have time. So, but every single one of them has a protocol, and we put it on the Open Science Framework. And now we'll probably put them on medRxiv too. Yeah, for sure. Because that's actually really important, because you'll see that there's also, with observational studies, that's one place where observational studies, right, you collect data that's retrospectively out there. You usually have models. You can move the models around a lot by changing variables, get results you want. So if you pre-specify what your model's going to be and put it in, it gives you a lot more credibility. I mean, I don't know how often when I review these kind of studies, I write to the editors like, confidential comments. I work with them all the time. I don't believe this one. It's hard to understand why they did this, and their model is probably data-driven. Data-driven meaning, like, again, you take your results, you let the data determine what you put in your model, and then the problem with those studies, they're probably not going to be able. They probably don't represent real life, right? So yeah, it's definitely a good idea to put your protocol somewhere. All right, so now we have a protocol and a plan, right? What about so now we're getting into registration. So this is different from a protocol. The reason registration is really, I think, only makes sense for trials. So when you're comparing interventions, or testing some kind of intervention, or evidence synthesis, systematic review. The reason, because the registrations are, like I showed you at the beginning in the school analogy, they're really short documents that give you an overview of what you're doing and that you exist, what you're doing, and later you can post results, what you found, but they're not very complete. The main thing about these, they're easily searchable. So it's not so easily, protocols are not so easily searchable. Now again, with medRxiv, they're going to become PubMed, but people put protocols everywhere. And so there's no, this one is going to, like for trials, if you go on to clinicaltrials.gov, it lets you search, and this is, someone mentioned before, Prospero. This is the Prospero for the systematic reviews. And those are basically the two places to register. I honestly don't think it makes sense to register. Other people offer registration services. I can't imagine, we'll talk about it in a minute, but why you might register anywhere else. So how did registration come to be, right? Why did we start this? This is just one paper. I'll give an example. So clinical trial registration was started as first. So, you have to list your trial before you start. And that started, the Americans started that around 2006, I think they launched it. And this clinicaltrials.gov. That's there are other, I'll show you in a minute, there are other registration websites. We don't have one in Canada, but that's the American one, the biggest one. But what was happening, it was becoming abundantly clear, particularly in the world of, the focus was on the pharmaceutical world, that many, many trials were happening. They were being launched, they were disappearing completely. They were going, the trial would happen, people may or may not know about it. People know about really big trials, less so about other ones, and then they'd just be gone. Any ideas what probably happened to these trials? Null results, negative results. They didn't find what they were looking for, or they couldn't recruit. I think that's if you can't recruit, then people need to know that too, right? Otherwise, someone else is going to launch a trial and try to recruit and spend a ton of money not being able to recruit. You still got to pay for your infrastructure and your team and everything else. So most of your costs are still going to be there. So Eric, we've had a chance to work with him a little bit too. He used to work for the Food and Drug Administration in the U.S., and he realized what was happening here. So Eric put out this classic paper in the New England Journal and he was looking at antidepressants. And what Eric realized that if you want to get your medication approved in the US, you do have to register a protocol, but not publicly, but with the Food and Drug Administration ahead of time that specifies how you're going to analyze your data, the whole shebang, right? And they're going to review your results based on that protocol. What Eric realized is that a couple things. One is that a huge amount of these protocols and trials disappeared, and that one, when they got in the literature, they didn't look at all like the protocols. So I won't go into this ad nauseum, but what, Eric looked at about 74 Food and Drug Administration registered antidepressant trials. You'll see at the bottom here, this graphic here, based on their actual protocols, the Food and Drug Administration, 38 of these found positive effects. 36 found null or negative. Negative meaning like harmful effects. Most of them were null. Look what got into the publications. 48 positive and 3 null. And even the null ones, I think, were spun, which I'll talk about later, meaning they didn't find an effect, but they made a case like this, adverse effects were less than someone else's drug, or something like, they were usually like something oddity like that, like someone wanted to make a point. So the drug companies were using the medical journals like almost like advertising literature, And how does a null trial become a positive trial? Well, they would only report some outcomes. They'd only report data from some centers and not others. They'd work with it until they massage it to be a positive trial. So the picture that we're getting was completely different. So this was just one example. It was one that clearest was a brilliant, after the fact, it's like, oh, great, this is great. But at the time, it was just a brilliant idea what they did. And back then, it wasn't so easy to, going into the balls of the FDA and getting those data in the early 2000s wasn't as easy as it is now. So it was quite an accomplishment. And they showed this. And so, a couple things happened at the time. So the Americans started this, the clinicaltrials.gov registry, where you can register your trials. And then something called the International Committee of Medical Journal Editors. And these are basically the general medicine journals, the major general medicine journals in the world. So New England Journal, Lancet, British Medical Journal, JAMA in the US, the Canadian Medical Journal is a member, and the Netherlands, there's about 12 or 14 of them that formed this committee. And they issued a statement that their journals would no longer accept trials that weren't registered before they started enrolling patients, as around 2006. And so these two things together have kind of transformed the idea that you need to tell people what's going on, tell people the basic aspects of your trial before you start it, or we won't touch it and publish it. And now, and for the pharmaceutical aspect, there's also, in the US, there's also some legal aspects to it too. Like I don't know how well they're enforcing them, but if you doing a pharmaceutical trials and you don't register, or you register and don't publish results within a certain amount of time, there's some fines involved. So, yep.
In other words, is anybody checking on the registered sites to make sure that those studies actually do get published?
Well, yeah, some people have done research on it. And no, they're not all getting published. I think it's better than it used to be, in some. But the other thing that's helpful too, though, is if you're doing policy and doing for evidence, you know when they're missing trials. At least. So you can't.
No, like you know that trials been registered, that's great, but it doesn't prevent, like, the other side of things, how many are not?
Yeah, I don't remember the number. It's come down from I think 50%, not a massive amount down, but at least that way you know that they existed, right? You know they happen. So it's better than what we said before. And we did talk about recruitment issues. We did a study on mindfulness, and I actually like mindfulness and do mindfulness, but I think I need problems with mindfulness. But I think that some of the evidence on it is, well, we publish it entirely implausible, right? It's impossible to publish it. But we also find that if you go there where we did that study, lots of mindfulness studies disappear. And I think, for instance, I suspect, I don't know if it's null results or also difficult recruiting, right? Not everybody's willing to do meditation, so which is important information. But at least you know that. And there is evidence on how many get published now. I just don't know it off the top of my head. I know I've seen these studies. So. And if you look at, quickly I'll show you what goes into a clinical. I'm not connected here. Hang on a second. I can show you what goes into the registration. Yep.
Would you suggest as well just doing scoping review and review open science framework because there is no.
Yeah, so I would put your protocol, but you can't really register it.
No.
Like here's the other point. The question is, should you register a coping review, I think, or publish it? I would put a protocol for any kind of study. Prospero for systematic reviews will only take reviews with health outcomes. And I believe they don't take scoping reviews. They will not take a review that looks at methods. You have to have a health outcome to register it for searching. And then, you know, for clinicaltrials.gov, they'll take pretty much anything, but I just don't think it makes sense to register something that's not a trial because no one's looking for it here anyway, and it's not set up for this. I'll just give you a quick, this is just one of our trials, the COVID trial we did, and show you what's there, but they've gotten a lot better actually, and they can't make you publish, but when the time goes by now, they're pretty meticulous now. It's amazing how much resources they put into it. They come back to you and they want the results, they want updates, so they do follow up now, which is pretty remarkable when you consider how many go through here and what's involved, and they're frustratingly detailed sometimes about the formatting and actually the quality control. I say frustratingly just because this is set up for a pretty traditional trial, and we do things like feasibility trials. Putting our round feasibility trial in their square peg format isn't always easy, but you know, if you go there, it gives you a summary of what the trial is about, right? You register, it gives you some more detailed description you can upload there. You don't have to upload as much as we do, but that's there. You know, it gives you but it's very basic, right? There's not as much detail. It gives you the basic eligibility criteria. So it doesn't tell us, like in our protocol, we'll tell more on how we assess each of these things, but it tells you what the basic criteria are. It tells you the basic study design, right? You know, what the intervention groups were. Again, not as much as the protocol, but it tells you a lot of information. And this is really important. What is the study measuring? Here's our primary outcome measure, the description, and the time frame. It didn't used to have all that there, but it's there now. Yep.
I have another question. You say you register a trial with primary outcomes and then you have a bunch of secondary outcomes and you want to test moderator and so on. And so you conduct the trial and you want to publish,
Yep.
And perhaps you want different students to work on this. Do you think it's ethical to split the paper, let's say one paper in primary outcomes, the other one secondary outcomes, or is it like more transparent to report all your outcomes from your trial in the same paper?
I think it's need to be transparent, so it depends, because some of your secondary outcomes are at later dates too. So you can't publish them all. I mean, there's no way you want to wait for all your outcomes. If you have outcomes at 1 year and then you have 3-year outcomes, so you can't wait, you can't publish, you don't want to wait for 3 years when your primary outcome's old, right? So I think all you can say is that, you know, we report all moderator outcomes are being reported elsewhere. But unless you think because moderators are more explanatory. So you can, just note that they're being published elsewhere and move forward,
Thank you.
Yeah. I mean, the thing is, if you have missing out, if you say here's our primary and secondary and they were all assessed together, they're not moderation, they're not explanatory, and they don't come into your paper, as a reviewer, I'm going to think that there probably were null and you're hiding them.
Yeah.
So you need to explain that, right? If your primary outcome gets switched or goes awry, you know, someone like me is going to throw a fit, and my wife will tell me to stop complaining about these papers and hope, you know, anyway. But it shows you your outcomes, right? And it doesn't show you everything. You don't have an analysis plan here, and there's still room for shenanigans, right? Like, you can still analyze the variables somewhat differently and put different models in, but it's a lot to go on, right? You know when it was registered. There's a date on it, and it also, it tells you. At this point, there's results. They become much more meticulous. There's an overview of results, right? So I can go down here and it starts giving you numbers from our study, right? A finished study. So it's a really great. And that's just one. That's the, I think, easiest to use and best administered one. We don't have one in Canada, so I can't really imagine. There are others. Like this is just the American. Oh darn! I'm not. Hang on a second. How long ago did my slides go off? Just now or before?
Just now.
Okay. Hang on a second. I just have to give it permission to share again, so hang on 1 second, sorry. Okay, so I clicked on it again, and it says ready to share now.
Okay, just click on this button.
Oh, that one? There you go, thank you. I always feel really silly in moments like that. You just have to touch it. Okay. All right. So yeah, this is, the one I showed you is the American one. I mean, there are lots of registries out there. They're usually national. Like there's a Dutch registry, there's a Chinese registry that are fairly prominent. There's an Australian, New Zealand registry. Given that we don't have one, it's hard to imagine why, just like the American ones, where everyone goes to search. The World Health Organization, if you're actually doing synthesis, by the way, has a compiler of registries. So you can't register with the World Health Organization, but they bring in all the national registries, and it's not as easily searchable as clinicaltrials.gov. You know, if you're doing evidence synthesis and want to find trials, we search both of these. So we search, clinical trials is a better searchable in the World Health Organization. But, so, you know, I think one thing that, yeah, question. No, I don't. I think there's no reason to register on more than one. Just register it once. And again, there's one. I mean, I think there's the American one, the clinicaltrials.gov. Biomed Central, the BMC journal, actually have one that they use a lot in the UK. I'm not sure what the relationship is between UK research institutes and that platform. And then everything else is national. So it's kind of hard to realize, you know, why just clinicaltrials.gov is the most prominent. I don't think I've seen Canadians register in other places. I think that's the one usually. But just to come back to, like, you know, this started out in, because there's so many, pharmaceuticals have such big implications and big money. And so they got in a lot of scrutiny. The registers were started really because of concerns about that. But I don't think they're bad or good people anywhere, you know, across, you know, people, this guy, one guy I've seen presenting on research studies and faking trials says, oh, these countries, they fake their trials here. We don't do it here. Well, no, we don't do it here because people anywhere, I think there's some people who will cheat and some people won't. In Canada, if I fake a trial, I'm gonna get fired next week pretty quickly. But I won't get fired for changing my outcomes. No one's gonna touch that one. My department won't be very concerned about it, unfortunately. In other countries, there's many fewer resources, there are many fewer infrastructures, there's a ton of pressure in financials, you know, China and others, financial incentives to publish these things. So their trials get faked. The people aren't any different anyway. But they said, okay, so it's also not, I had just, you know, a student took a job recently in industry and said, oh, I feel so bad. Like, no, industry does great things too. But we're not any better than they are. In fact, I think we're worse right now in many ways. So they're regulated and non-regulated trials. Regulated things that have, like the Health Canada, the Food and Drug Administration, our drugs. There's also medical devices tend to be to some degree regulated. Most of the trials that most of us do here are psychology, physical therapy, surgery of all things, right? None of these are regulated, so non-regulated. There's no legal implications for registering your study. But most of the journals, or many of the journals say that they're gonna require registration, right? But we did this was a crazy study too. Marlene Assar, who's just finished med school here at the UDM, led this study. So we went on live. Every day we pulled all the trials out of PubMed in all the journals in a series of, all the journals and fields that publish non-pharmacological trials. You know, nursing, psychology, physical therapy, all the, you can imagine, there's a whole list of them. And in a couple of months, we pulled out 953 intervention trials. And we want to do it live because we looked quickly every time they came up, what was the journal registration policy on those days, and did they follow it, and were they registered? And sadly enough, as you'll see here, we had publications from 254 journals, but only about 10% required prospective registration. This was only 5 or 6 years ago now. So most of them didn't require it. And you'll see that only 20% of the trials were registered ahead of time. And registering after the fact isn't helpful, right? If you've already seen your results and you put it on the registry and you tell me your primary outcome was X, well, you already know that what happened with X. It doesn't help me verify what you've done, right? But, so only 20% were registered a priori. But then the problem got worse from there because there were only 17 journals that required prospective registration before you enroll people and had at least 2 trials, none of them had at least 50% of the trials actually registered a priori. So nobody actually followed the policy, not a single journal. Now, why do they do that? I suspect these are specialty journals. They're in our fields. They're psychology journals. There's some benefit to having a perception of being rigorous, but not holding our peers to it, actually. It's a complicated issue. I tried to get some economists here to do game theory analysis of this, and he didn't want to. I thought it was a good idea. But anyway, it's why that happens, but it happens, right? We're not. So, but even worse, we trace back out of these 953 non-regulated, right? So psychology, mental health, physical therapy. How many can we trace back a primary outcome that was the same as same primary outcome published as the registered one? 3% of the trials. Because either they hadn't registered a priori, so that's 80% right off the bat, right? And then the rest of them, either they register like 10 primary outcomes. Your primary outcome, if you remember, you get a 5% chance at a false positive. You either need to register one outcome and say, that's my 5%, and other ones are more secondary. Or if you have 2 primary outcomes, you need to split up your p-values, right? And then a lot of these just change the primary outcome. They said, well, it was changed after the fact, and almost certainly because it hadn't been significant, but another one was, right? So, so we have a lot of work to do in this area. So, and I think one thing important I want to mention here that when you're doing your studies, null outcomes are really important. We have this ethos in medicine that or healthcare, and that your job is out there to find solutions to people, and you must find one. But you're not helping people if you manufacture one that didn't happen. In fact, you're misleading people into doing things that don't help them. You're misleading people into going down trails to verify your work that probably can't because it didn't happen in the first place, right? And it's a real challenge here. So, I was lucky to have a good mentor who said, stand your ground. This is right. There's a lot of pressure on you to publish things that, it's much easier to publish a result that says, if you go in a psychiatry journal, to publish a result that says everything we're doing is great. These are all members of our guild, right? But it's really, really critical. Null outcomes are really important. They're not going to get you as rewarded as highly, unfortunately, but it's really critical you get them out there. We need to know about these, right? As an aside, our team, I think it's funny, our team publishes fairly often in JAMA and BMJ. We cannot get into psychiatry journals, ever. We always get rejected in psychiatry journals, which I think reflects a little bit that, not bad, but the specialty journals tend to be a little bit more like, what is your message, is supporting how well we're doing. And we do great things, so I'm not a nihilist. I think, there's great work going on in mental health, but we need to be able to publish all the outcomes, and again, it's critical. So coming back to that, so really, if you come up with a negative outcome or a null outcome, publish it. Go forward and report it. That's really, really, really important. Okay, so the last stretch here, we're going to go into now reporting guidelines, right? So I want to thank David Moore, who shared these slides with me, but not thank him for making them pop up one at a time. I hate that. I didn't realize that was, I didn't realize that was coming that way. So anyway, so how can our reporting, now we're going on to reporting and publishing what you've done, right? You've done your study, you've put together your protocol, you've registered it if it's a trial or a systematic review, and now you've done it. David has this taxonomy of poor reporting. Obviously the top one, if you don't report your findings at all, you're killing us, right? Because we don't know what happened. Selective reporting of outcomes, again, that's when if you collect 10, if I collect 10 outcomes and only 2 are significant, I tell you that, if I tell you I collected 2 and they're both significant, that sends a very different message, right? So selective outcomes is a huge issue. That was what happened in the slide I just showed you of our study, right? I think a lot of people changed their outcomes or didn't report the negative ones. Selective reporting of analyses done. You can analyze your data in different ways, right? I mean, and some of it's not due to people being bad people. Some of it is people are cheating and know they're cheating. Sometimes people just don't know, right? Like I've had people come up to me and say, we've analyzed our data using this model, it's not significant. Could you help us find a way to get it significant? And my response is, no, you have to publish this. That was your plan, right? Because they're clearly not aware that what's happening and how they're misconstruing their evidence. But you can, analyze the data the way you said you were going to analyze it. If you don't think it's the best way and you're going to change the analysis plan, tell people that you've done that and why, right, in your report. Interpretation bias or spin, and I'll come to that in a minute, what that means. Inconsistencies across sources we see all the time where the protocol and what you say you're going to do in the protocol and what you say you did in the publication don't match. And the big one here is this incomplete publication. This is the most relevant, I think, as you're reporting and using reporting guidelines, that, unknowingly, you won't have information that a good guideline group or a policy group or a systematic review will need to be able to use your study and credit it for being a useful source of information, right? Again, I showed you, like in our systematic review on COVID, how much evidence we pretty much disregarded in some ways, right? And then even at the guideline level, how weak the guidelines are, and because we don't, most of it's because we don't know. It's reported so poorly, we just don't know what really happened in the study. Okay, so what are reporting guidelines? They basically have three components. There's a checklist. There's a flow chart of what happened, how people, or how in a systematic review, how studies got into your study. And this is the last one's really important too. There's something called there's an explanation and elaboration that's going to describe all these things. And I'll talk about that. Those are three parts. And good reporting guidelines, and I'll show you where to find them through the Equator Network. They're things that are evidence-based in themselves. And I'll show you the process for getting them, but they're really vetted quite rigorously. There's different people, stakeholders giving input. They're carefully developed and tested. And they use evidence to inform what they're telling you should report. But a reporting guideline is supposed to be the minimum set of information that you should have provided in your study. Now, the nice thing is, too, is that some journals you'll submit to will say you get 3,000 words. I mean, CMAJ gives you 2,500, for God's sake. So how could you actually report these things? The nice thing is that, again, you can put anything in an appendix. And like the systematic review that BMJ doesn't have a word limit, but after our COVID review, they came back and said, too long, cut it to 4,000 words. We were at something like 7. We dumped our entire results into an appendix. It was very detailed, and then we wrote a summary, right? And the methods is often that way. You can put a kind of a summary, a short version of your methods, and all the detail that you need to meet these things can be in a full methods in an appendix. Even if you have your protocol, dump your protocol in the appendix, and you've met these reporting guidelines, right? It's there with your paper. So this is just an example. This is the Prisma for diagnostic test accuracy studies. It could be anyone. But you'll see there's a fairly short, it's a long, there may be 30 items. And it goes through each of the steps of the paper. It's nice, it's a little roadmap. Like when you're writing your paper, pull it out and make sure you're writing these things in your draft. And then, in our group, we're fortunate enough that we have resources and staff. We have, in addition to the ones of us who wrote a paper, someone on our team who didn't write the paper goes through with the reporting guideline and checks and makes sure we have everything set up. We double check these things. And don't get fancy here. And no one's reading your papers, and few people read your papers from front to back. It's not like a novel, right? So people read these things differently. People need to know where to find these things. So put them where the reporting guideline says to put them. As someone who's methodologically oriented, I'll probably, if you submit a paper and I'm reviewing or even reading it, I'll probably look at your abstract, go to the tables, maybe the parts of the methods, and depending, I may or may not read your introduction and discussion or something. If I'm reviewing it, I will, but otherwise. Other people, some clinicians might read your introduction, abstract, introduction, maybe your discussion. Other people, but people need to know where to find things, right? So don't be clever, don't move. One of the biggest areas I find, particularly in the mental health area and psychology, we put results in the methods, right? We put who our sample was in the methods and things like that, right? Because that's a result of your method. You didn't. Everyone can't get the 432 scleroderma patients you have as part of your method, so that's your result. But in any case, it's a nice roadmap, and it's not that hard, to go through. It's also good how to learn how to write a paper as a guide, an overall template, how to set this up. It's really useful, okay? You know, they all include a flow chart, and this is just really basically part of the reporting. These are complicated ones, but they're flow diagrams, but look through the guidelines, they all have an example. I mean, a simple trial, the one on the left, is a systematic review, but it's really nicely done because it shows how you can get information from a prior review that you use and build upon, from your database searches, from other evidence that people gave you, but someone should be able to track back where you got everything and count, right? Where your studies came from. If they can't do that, I think you're dead on arrival, right? If you can't replicate that for a good journal. And this one here is a trial, you know, but it basically this is really critical. These are really critical issues. How many people were actually eligible for your trial? How many participated? If you got 70% enrolled versus 10%, it means your trial is more generalizable, right? If most people who were eligible signed up, did it. And then the follow-up, right? How many were lost and why they were lost? So these are things that people want to know. These are one of the biggest reasons people downgrade evidence and don't use it, that you lost a lot of people in your study, or you didn't use analyses that could account for that, or whatever it might be, but this is a critical part. So there's a flow diagram. Oh, and then there's a explanation elaboration. These are long papers. Like this is just, this is one of the ones I was involved in, the PRISMA is systematic reviews and meta-analysis for diagnostic tests. And this is just one example from one item. It's just part of the item. I didn't put the whole thing up here. But these are nice because it tells you what the item is. It tells you what you're supposed to report. So this first part over at the top by the orange box is actually the item, right? You know, state the principal diagnostic accuracy measures reported. That's it. That's the item. That's what you get in the checklist. But then they give you some nice examples there of how people have done this, actual quotes from papers, and then they explain it here. And this is just part of the explanation. There's a real tension among the guideline developers because some of them someone always says, these are not textbooks on how to do methods. We're not teaching people the methods. That said, they're pretty good textbooks on methods. So it's fair enough. You can't learn to do these studies from these things, but you can learn a lot from these explanation papers. So, you know, especially if you're getting started, the first time you do one of these papers, one of these designs, and these reporting guidelines are set up for each kind of trial design or study design, okay? And I'll show you in a minute. But these explanations and elaborations provide a lot of information on why people do things the way they do. And they're really informative. Like I learned a lot when I started to do research and reading these explanations. They tell you things that, you know, again, yeah, why they're doing things this way. And these are developed, you know, David, okay. Again, thanks to David for the slides, but no thanks for how he set these things up. Anyway, you know, they're quite rigorously developed. So we did one, we do in our SPIN, our scleroderma cohort, we actually set up a cohort so we can use it to run trials through it very effectively. That's one of the reasons we could launch a trial in 3 weeks in COVID. We had a standing cohort of people all the time. We just had to plop in and do it. There's no new recruitment, right? So we did one of these consort guidelines, which is consorts for trials done in cohorts. And, but, you know, how do you get these guidelines? They're not just what a group of people thought, right? So the ones that are on Equator, which I'll show you, are really good. There's some other ones like Joanna Briggs does evidence synthesis, and they do some good things, but their guidelines are not well vetted like this. They tend to be what a few people around the table thought. But the ones that we're talking about here, you know, what happens here is that, you know, we get CHR grants to do these, or other people get them. You do evidence synthesis on, so if you're looking at, you know, trials done in a cohort environment or clinical trials generally, the group that's done these guidelines has gone out and looked at best evidence on what should be done and why things should be reported, what's important information, what could lead to bias or not. They do, once they do that, they've come up, you know, a general preliminary list of items. And then there's a Delphi process. Delphi process is basically expert opinion, but like for our reporting guideline, we went out to about 120, I think, people who have done trials, and it was cohorts or other registries and existing data structures. About 120 people who vetted each of the items gave their opinion on how important it was to be in the guideline, and then rationales why, other examples. So it went through 3 rounds of that. It went from about 120 the first round to probably about 80, I think, the last round. But we got a lot of feedback along the way. It wasn't just your opinion. It wasn't a vote count. But people were knowledgeable people provided explanations on why, and we made a lot of changes because of that. Then there was a consensus meeting. We got to go to London to do that. Now they're all on Zoom, right? But we all met together, and then we got in a room, and and it was a room of about 25 people that were really good at what they did. We had a couple patients there from our network, the BMJ editor, methodologists, trialists, all sorts of different kind of people, different perspectives. And it was a nice environment. People really held each other's idea. Are these really necessary? Why? Based on what evidence? Are they unique to this kind of study? And really hashed out what should be there. And then after the meeting's done, these things got tested, right? We pilot tested our actual checklist and had people use these and provide us feedback on using it before we did the final one. Anyway, but the reporting guidelines are, they're a guide for you putting together a study and reporting it. They help people appraise, how useful it is what you've done. Like in peer reviewers should ideally, if you're ever peer reviewing, you should always pull out a reporting guideline and look and review it based on that. And it puts everyone on the same playing field, right? And this is me because I think it's probably always been a problem, but increasingly as peer reviewers, I've mentioned, are under pressure. I think the quality of peer review has gotten pretty awful in many cases. Not always, some are very good, but they can't find reviewers. They're going farther and farther, and so sometimes you get more and more ad hoc opinion-based comments. So these are actually, not only do they help you report your study, they help you go back and say, reviewer number 1 has indicated that we need to do this. The reporting guideline says X. We have done it in consistent reporting guideline. We trust that the editor will, whatever. And so they actually put you on. They have a set of rules that you don't have to just slavishly obey the whims of someone out there, right? Because you can justify. So where do you find reporting guidelines? The Equator Network, it was set up, David from Ottawa, Moore, and Douglas Altman, who's now deceased, from the UK, and a group from France, are some key players, and Isabelle Boutron, Philippe Raveau, and others. Now it's a big network, put this together. But this is an aggregator of reporting guidelines, and they're the ones who set the standards on how to develop reporting guidelines I showed you. They've done just a remarkable amount of things. There's toolkits and library. This was a great website for learning all sorts of things about research, and it helps you find reporting guidelines. The main ones here, so they're based usually on study designs, although there's some that are specific to areas. Like acupuncture is going to have specific things because there's certain nuances about acupuncture and delivering it and blinding it that need described that are just specific there. So there's an add-on, right? So you'll see here the ones I've highlighted. Consort is for trials. I'm terrible with acronyms. I have no idea. They're long names that go behind these acronyms. But STROBE is for observational studies, so cohorts, cross-sectional studies. PRISMA is for systematic reviews and meta-analyses. SPIRIT is for protocols for trials. START is for studies of diagnostic accuracy. Those are the main ones. And then the SRQR and CORAC, there's 2 that are usually used in qualitative research. I think CORAC I see more often, is probably more rigorously done in my opinion. But those are the main ones to start with. And then there's some extensions. What does extension mean? Like if you're doing a trial in a cohort, you use the consort for trials and look at our extension for some other items you've used. I mean, you'll see there's these are some of the things I think that are, I'm assuming a lot of people here are going to use these ones, but oops, there we go, okay, hang on a second. All right, these are some ones that you might find particularly useful for a lot of work you're doing. Isabelle Boutron, she's one of my favorite researchers. She's just fabulous. She's out of Paris. She did the non-pharmacological treatments one. So a lot of you, if you're doing anything that's non-drug, it has a lot of elements that should be reported there. This next one on the left tide here is great. So it goes through intervention descriptions. Danielle Rice, one of my ex-doctoral students, did some work in Ottawa with David's group, and brought this to us, said you're not following tide here. It really goes through what you need to be reporting about the interventions you're using really nicely. And again, if you can't fit it into the text, put it in the appendix, but it's tidier is a really good one. There's so often in our reviews, we don't really know what the intervention was well enough for certain aspects, and we can't use it. We use it, but we downgrade how much weight we give to it. So tidier is a really, really good one. It helps describe your interventions, the different elements, what people want to know. You know, things like who was delivering it, how were they trained, what the material was that was people received, exactly what the timing was and the length, all sorts of things that you may not always think of. Consort routine, that's a little bit, those are for trials and cohorts. I think I put it there because of my ego, because we did it, but that's, and then, the Consort Pro, patient-reported outcomes, is really helpful. It's specifically focused on patient-reported outcomes, so not like mortality and hospitalization. Anytime people report things on a measure or whatever, what you should be telling us about the measures, the validity. That's really, really helpful for any, it's set up for trials, but any kind of study, it's a really good resource, and so forth. Now, you'll see, like, if you're doing one of these, if you're starting with a trial, you'll use Consort, and then there's some extensions to be used also. So how do you figure that out? It's a little bit messy right now, frankly. I mean, you've got to ask somebody, you know, you can look on Equator and search it too, but you want to probably consult with somebody who knows. Like, what are the applicable reporting guidelines you should be looking at? This group, this is Isabelle and Philippe Ravaud from Paris. They set up this cobweb. It's a nice thing to look at, but it amalgates at least some of the reporting guidelines. So if you give them the basic, you know, characteristics, it's a non-pharmacological clinical trial with a couple things, they'll pull those together and give you one template to work with. It hasn't been updated as much as would be ideal, but you can look at that. It might be it can be useful to figuring out what you want to be reporting. Again, this is just a reporting. The website has all different things you want to look at. It has a library of different resources that are not just reporting guidelines that are useful. This is, and then finally, one of the last things I'll get into, and coming back to Isabelle, she's a fan. She's just fan. She and Philippe Ravaud, they do like clinical trials on things like reporting and clinical trials on tools to help people report better, and they're really creative. They're really good. But this was a study that she did. She introduced the word SPIN, which is ironic because my organization, the scleroderma organization, is called SPIN. So SPIN is defined as reporting that distorts interpretation of results and misleads readers. That's not what our organization is. You know, scleroderma organization, we're not trying to mislead patients and doctors. But Isabelle, to the best of my knowledge, introduced that term with this paper. In this one, she looked very specifically at what people were doing when their primary outcome was null or negative. And they went through and meticulously documented how often people were not putting the primary outcome in the abstract, were changing it, we're really saying, well, it wasn't significant, but it would have been if, and having all sorts of caveats. It basically spun their papers into a null or negative paper that looks positive. And there's been more papers since, and I made my plea for this before. You know, it's really important that stick to your guns. Null findings are important. There's a lot of pressure not, to report things differently. We did a trial on a hand rehabilitation program in scleroderma, and we tried a new trial model where you basically you randomize people when you offered it to them before you knew if they wanted it or not. There's a number of reasons for why you might do that, and Isabelle worked with us on it. I'm not sure why my slides are advancing. That's David's fault too. Anyway, I tucked a couple of the slides in, everything went awry. Anyway, we did this trial, and what happened in this trial is that it was a good trial methodologically. We understood some pitfalls of this method, but people didn't accept the offer to use intervention, so people didn't use it. When they did use it, they didn't use it. When they accepted it, they didn't use it. And our result was almost significant in favor of the intervention. And we reported very clearly, it was no, it didn't work, nobody used this thing. And we need to rethink how we do these trials. We don't know what would happen if people actually use this intervention. But I remember one of our colleagues said, you need to write that with a few more people, it could have been significant. You have your effect size, a few more. It's like, well, no, no one used it. I think it was actually an example of reporting of how like non-blinding can influence results, because the fact that nobody used it, we're almost statistically significant. Like, come on, they didn't use the damn thing. It didn't help them. So anyway, but it's really important to stick to your guns. There's a lot of pressure to twist things and describe things. The other thing I see as reviewers sometimes, you read discussions, like that tell people also, they find a trial, they do it, it didn't work, and they're writing this whole thing, well, it didn't work this time, but we know it, basically know it works, and here's how you should use it, and here's what you should do. And my comment as your reviewer is like, well, why did you do this study? You know, if you believe your study was important, then write that it didn't work, and maybe there's another trial to do, another iteration of trying it, but don't tell people to use it anyway. And the last thing I want to touch on, and I'm not really an expert on this, honestly, but it's really more increasingly important that to make sure your data are available after your trial. That's part of transparency and completeness now. It's getting easier and easier, because for a number of reasons. And this is just a McGill site, but I know that UDM has a site too, and people that do this. All good institutions have people working on this. But after the study, make your data available. There are a number of reasons for that. You know, people should be able to verify what you've done. Someone should be able to say, I want your data set. I don't trust what you did. And that's fine. They should be able to vet it and verify it, right? And it's also for data sharing. I gave the example of the individual participant data meta-analysis we do, where we bring in people's data, we put them all together, and that lets us do things like not only just aggregate the data, but we can run subgroups that no one else, a lot of studies are like that, you know, like for sex and gender. Trials are often not big enough to lose analyses, but if different trials do have those data, you can bring them together and analyze separately by sex. So it's really critical that your data are available and warehoused in a way that they're, you know, there's this group at McGill, and I'm sure again UDM clearly has this. I don't have the site, but have people that are expert now on how to do this in a way, how to go, what your consent form should look like, so you make sure you can do that, right? So what the permissions are like. And you know, and we have McGill has something called Dataverse, but it's basically, again, it's a data archiver, whereas there is set up in a formal institutional warehouse for data, not just on your computer anymore, which depends on if you leave the institution, if you go somewhere else, you know, you don't have the data. And there's a lot to think about. It's not as straightforward as you might think. So some people advocate for all data should be available for everybody and period, put it out there. So we don't actually do that. We actually put our data here, but like for our individual participant data meta-analysis, we can't give it to you. The reason we can't give it to you is because we don't have ethics approval. We bring in data sets from, we have like 300 institutions. We don't have approval to reshare. So we can't share it broadly, but it is there. And if someone, we could, I think with the ethics, if you wrote and said, I don't believe your results, I want to rerun your analysis, we could set up a contract to allow you to do just that and do it that way, but we couldn't, there are limitations, right? And honestly, for our scleroderma project, we warehouse our data here, but our policy there, we go through a lot of effort. So anyone in our network who uses our data for papers, they have to submit a protocol. They have to register. We don't let people get our data. We don't let people get it. We're the central hub. We don't let people have the data until they've done these things. We want to ensure that we're not going to vet your paper based on what you find, but we are going to vet your paper based on that you've done the right things. And so if you make an entirely open data set, you lose control over that. So I don't know if I'm right on this one, but we've taken a position that we will give you, if you want to use our scleroderma data here today and you write me, you can. We'll allow you to have it, but you do need to go, first you need to submit a protocol. We have our statisticians look at that, not to block you, but to make sure you have the support you need to do what's helpful or they think if you want, and support you to do it. But you can't just get our data set and spin the dial as many times as you want for analyses until you find something clever. So if we're going to put our own people through the steps of doing the right thing, we're reluctant. I don't know if that's the right answer. Some people out there would say that's the wrong answer. Your data should be available publicly. So I think that's an open debate, but that's how we do it. So anyway, that's my best spiel on and I think how you can do your best to make sure that when you're doing your research, you're doing all this work and all this time and all the stress you go through, that you document it as well as possible to make sure that others use it, that you get credit for it, for doing the good work you do, and that it gets uptake, right? That people make policies based on it or integrate it in other or systematic reviews, and that it doesn't stop because you've published a paper somewhere and it's done, right? So thanks a ton. Thanks for the invitation, Claude. I'm really happy that you invited me, and thank you for listening. And I'm happy to take questions and comments or talk to anybody after too.